Hello everyone. This is the second part of lecture 1. In the first part, we learned about Coase postulate and its limitations. And in this part, uh, we'll discuss in little bit more details about the postulates and limitations with example. So let's just start. So the first postulate, association of microbe with lesions of the disease. Okay, so before proposing the rules or postulates, Robert Koch, he was able to establish the connection uh, between. He was able to successfully uh, identify the causative agents of anthrax and uh, Mycobacterium anthrax and tuberculosis. Okay, so that means he was able to successfully identify, he was able to successfully make the connection between Bacillus anthracis and the disease anthrax and Mycobacterium tuberculosis and the disease tuberculosis. Okay, so for him, establishing the connection between anthrax and Bacillus anthracis was easy because at that time there was a system to culture Bacillus anthracis. It was easier, and also Bacillus anthracis is easy to detect by staining methods which were available during that time. So he easily established the connection between Bacillus anthracis and anthrax. But while he was trying to find out or identify the causative agent of tuberculosis, it was actually not that easy. I mean, initially, Koch was successful in saying that if samples is isolated from tuberculosis from different animals and re-inoculated them into a specific host, the host developed pneumonia. Okay, Koch was able to show that at that time, but yet he was not able to firmly say that the Mycobacterium tuberculosis bacteria it caused the tuberculosis, because at that time there was no method to culture the Mycobacterium tuberculosis, and also there was no staining method to identify the Mycobacterium tuberculosis. So first he has to first Robert Koch, he has to develop the system to identify the Mycobacterium tuberculosis bacteria. He developed a staining procedure, a special staining procedure by which he was able to stain the Mycobacterium tuberculosis. Then he was able to successfully demonstrate that Mycobacterium tuberculosis it caused the tuberculosis. Okay. Now, okay. So from this experiment, we can say that okay, if we can invent or develop a identification system to identify microbes, it will solve our problem. Well, it's actually not that simple. So, for example, let's talk about the Helicobacter pylori. So, people know that Helicobacter pylori it colonize in the gut of human. Okay, in the intestine of human. People know that. Okay, so although it is still not very difficult to culture Helicobacter pylori. Okay, so nowadays we can culture it with difficulty. It's still not that efficient, but yet we can culture at least. But there are other methods to easily identify pathogens nowadays, right? Say the molecular methods, PCR methods. Anyway, so while in case of Helicobacter pylori, it's actually a very common bacteria which is found in gut of many people. In especially like even in the developed country, almost like more than 70, around 70 to 80% people they carry H. pylori in their gut, and in the developing country, like say for example Bangladesh, almost nearly 100% people they have this bacteria in their gut. So scientists knew that. Okay, they were able to detect the H. pylori from the gut tissue, not by culture, but say by molecular method. People know that, but yet scientists I mean, the scientific community took 10 years to accept that H. pylori caused the ulcer, and you can easily guess the guess why, right? Because H. pylori is, it's kind of present in every person, but do every person develop ulcer? No. So some people they develop ulcer, others not. Okay, so that means even if you can easily detect the microbes in its site of infection, we cannot easily say that it caused the disease. Basically, these things make it clear that disease is not solely dependent on the pathogen itself. Other factors like the age, immunity, genetics are also necessary, are also important. Okay. So anyway, so after 10 years, actually, finally the scientific community accept that H. pylori caused the ulcers. So the scientists who were trying to establish this one, they were actually kind of frustrated because they knew that, I mean, they could assume that it caused the ulcers, but to prove that, they have to satisfy the cause postulate, right? So, I mean, they have to inoculate a host with H. pylori, and that host should develop ulcers. And there was no good animal model. Actually, still there is no good animal model to do this experiment. Okay, so one frustrated scientist, he used himself as a guinea pig to establish. I mean, he himself take the H. pylori and develop ulcer. Okay, he himself act as a guinea pig, and he finally the scientific community accepted that okay, H. pylori ulcer can cause ulcer. Okay, another problem is chlamydia pneumoniae. Okay, so this bacteria, uh, as its name is chlamydia pneumoniae, it can cause pneumonia, but it can also cause. I mean, it's assumed that I mean, oh, chlamydia pneumoniae cause pneumonia. That is, and in addition, people used to think that these bacteria cause heart disease, atherosclerosis. Okay, I mean, scientists used to assume that, think that chlamydia pneumoniae also cause heart atherosclerosis. Okay, why scientists assume that? Why they used to think that? Because in many cases of heart atherosclerosis, scientists were able to isolate chlamydia pneumonia from that disease heart. Okay, so from that they assume that. Okay, however, chlamydia pneumoniae is also sometimes also found in healthy heart. Okay, so scientists, I mean, so that's why it was a confusion, right? I mean, it is found in the disease heart, and it is found in the healthy heart. So now there is the big question: do these bacteria really cause that heart disease? I mean, heart atherosclerosis. Okay, so it's a big question. So there was debate among scientists, and actually finally it was found that these bacteria actually don't cause the atherosclerosis. Okay, I mean, if I mean a healthy heart is solely infected by only these bacteria, they I mean usually the atherosclerosis is not formed. Rather, scientists find out that I mean presence of chlamydia pneumoniae, it may increase the rate of atherosclerosis. Okay, I mean it may aid to develop atherosclerosis, but they are not the sole agent. Okay, so things are not that simple, right? So I think you can understand now that it's not that simple. So if there is a disease person or a disease tissue and we just isolate a bacteria from that tissue, it doesn't mean that that pathogen caused that disease. Okay, so we have to consider every other factors, every other parameters to say that okay, this pathogen caused this disease. Okay, so that's why satisfying all the cause postulates is necessary. Okay, step by step. Okay, anyway, then the second postulate, isolating the bacteria in pure culture. Okay, so we already know about this one. I mean, there are many pathogens which we cannot culture easily. Okay, so just one thing. I mean, whenever we say, especially for bacteria, I mean, culturing the bacteria. So, if we want to satisfy the course postulate, we need to isolate that bacteria from the lesions. That means from the diseased tissue, and have to I mean, should be pure culture them. Culture them in pure, as a pure culture. But I mean, whenever we talk about bacteria, it come into our mind that culturing bacteria means culturing them in the agar media, okay, or the media, okay. So, but the thing is, it's not necessary that, I mean, to satisfy the course postulate, it's not necessary that we have to grow the bacteria into agar media, okay. So there are many bacteria which don't grow in the agar media, okay. So in that case, but if that bacteria grow in like cell culture, it's okay, we can use the cell culture to grow the bacteria. And if we can grow that bacteria in the cell culture, it's still, it's same, right? The postulate was we have to grow them as pure culture. So it's not necessary that we have to grow that bacteria in the agar media. If we can grow them in say host tissue culture cell, it's fine, okay. So in fact, I mean, for virus, we can never grow them in agar media, right? For virus, we have to always, I mean, if we want to satisfy the course postulate for a virus, we always have to use the cell culture to cultivate the virus or grow the virus, okay. Okay, then limitations of isolation of microbe in pure culture. Okay, I just discussed about this, right? And we know the problem because many pathogens, many causative agents, we still there is no culturing method to culture many causative agents, right? So, but yet, I mean, so although there is no culturing method, but we have to establish the connection with a disease and a microbe, right? Because if we cannot establish the connection, we cannot figure out the preventive measures, the treatment, right? So actually, that's why it's alternates, or there are nowadays there are different alternative ways to establish it. Okay, to satisfy the course postulate. We'll talk about this later. Just now talk about the. I mean, let's try to understand the limitation first. Okay, so many bacteria or many microbes, there is no system to culture them. One example is Treponema pallidum. Okay, Treponema pallidum, this microbes or this bacteria, it cause STD, sexually transmitted disease, named syphilis. Okay, it's actually established now. It's very well established that this bacteria cause the syphilis. However, till now, no one was ever able to culture this bacteria. Okay, in pure culture. Well, there is a way to culture this one in an animal model. Actually, Treponema pallidum can be cultured using rabbit testicle. Okay. However, there is no method to grow it in pure culture. Okay. So we have talked about some of the limitations. So what we are getting from this, the thing is, there is limitations, but we have to try to find out a alternate. Okay. Well, so all the alternates we have to find out to satisfy the original course postulate. So keep it in this mind. I mean, the original course postulate has many limitations, but we are trying to find out the alternate to satisfy the original course postulate. Okay. Okay. Now the third postulate, showing that the isolated bacterium cause disease in humans or animals. We already know that it is actually a very difficult step to do, because for human disease, we cannot use human as a host, and also it's very difficult to find out a perfect animal model in which we can reinoculate the pathogens and develop disease. Okay, so one good example would be HIV, right? Because HIV only cause human disease. It's a human specific virus. Okay, so there is no way to satisfy the third postulate if we want to satisfy in the original cause postulates way. Okay. Okay, then limitations of artificial inoculation of microbe into a susceptible host. So same things. I mean, we cannot use human, and most of the time there is no perfect animal model. Actually, the truth is, for most of the diseases, actually very difficult to find the animal model, and that's the truth. There are very few perfect animal model to mimic the human disease. So let's talk about one example. Say Salmonella enterica typhi. Okay, we know that this bacteria, Salmonella enterica cerevisiae typhi, it cause disease in human. So the scientists who want to study with this disease, they never find out a good animal model. So to study the pathogenesis of this disease, some scientists they use uh, mice, but Salmonella enterica cerevisiae typhi, it don't cause any disease in the mice. So there is no way to study this. So, but still we need to study the disease. So some scientists do, they use a, you can say, a family member of the Salmonella enterica group, some like such as Salmonella enterica typhimurium, cerevisiae typhimurium. Okay, this cause disease in the mouse. So they use this bacteria, Cerevisiae typhimurium, to study the pathogenesis of, or to study the typhoid using mice. Okay, so this is actually a problem. Like, say if we want to, I mean, say now it is established the Salmonella typhi, Cerevisiae typhimurium, Cerevisiae typhi cause disease in human. Okay, but if something a novel disease occur, then it will be and a novel disease occur, and if we cannot find a perfect animal model for that novel pathogen, okay, it will be very difficult to satisfy the third course postulate, right? Okay, now the fourth postulate, re-isolating the bacterium from the intentionally infected animal. Well, if we are able to do the first three postulate, then the third postulate is easy to do, right? Because in third postulate we just have to re-isolate the bacteria from a intentionally infected host. Okay, so if we can do the second the second postulate, I mean isolating the second, what the second postulate, we have to isolate the pathogen from the host. Okay, so if we can do that, definitely we can also do this one, right? Okay, so that was the, I mean, about the postulates and its limitations. Now, so we know that there are lots of limitations, but yet we have to satisfy the postulates. So what we can do, so that's why there are some modern alternatives by which we can satisfy the postulates. Thus we can establish a connection between a pathogen and the associated disease. So we'll discuss about this in the next lecture. Okay, till then, thank you.